Hi there, I'm Glenn Morrison. I am one of the professors in the Civil, Architectural, and Environmental Engineering Department at Missouri University of Science and Technology. What you see behind me is the civil engineering building at the university. The environmental engineering group at the department, in this department, works on a wide variety of environmental problems and technologies, including fly ash, metal, transport into the environment, trees that absorb pollutants from soil, wetlands studies, landfill studies, and even studies about obesity in the environment. I work on indoor air pollution and indoor air quality from a chemical perspective. I study how pollutants like ozone and other contaminants in indoor environments interact with surfaces. The purpose of the video today is to show you what kind of equipment is used to study how chemicals are taken up and the rate at which chemicals are taken up by building surfaces. And what we mean here is that the chemical of interest here is ozone in this case. Ozone will actually stick to a surface and then react with the surface, generating potentially new compounds, but also sometimes just going away, basically decomposing into chemicals that we don't really care too much about, like carbon monoxide and carbon dioxide. The equipment that I'm going to show you and that my student is going to show you is specifically designed to measure something called the deposition velocity. And this is what's called a mass transfer coefficient that we can compare from one material to another. And we can use it to predict how a particular material or how much that material might remove a gas from the indoor environment. So for example, if you put carpet down that had a very high deposition velocity, that means that the indoor concentration in the air of ozone in that building would tend to be lower than putting in, let's say, vinyl flooring, which has a low deposition velocity for ozone. All right, so what we'll do now is we'll take a look at some of the equipment that we use for that. Okay, so this chamber here and the chambers adjacent to it are all temperature controlled chambers, one of which is actually a humidity and temperature control chamber for doing environmental engineering experiments and studies. They're convenient because you can put entire test systems into one of these and have them all at the same conditions. Let's take a look inside. Inside are a variety of things, including several chambers, some samples, and it's, as usual, a little bit untidy, but that's normal for these systems. Let's go in a little bit closer here. This chamber here is the chamber that we use for ozone deposition velocity studies. The one over there is used for a completely different set of experiments. Here are mass flow controllers. These control the rate at which air flows through the reactor. We can use this bottle of water here to humidify the air and adjust the amount of humidity in the air by mixing gases through there. This is an ultraviolet light ozone generator, and in combination, by adding water and ozone, we can get just the right conditions. These valves at the top are used to adjust whether we are measuring the upstream or downstream ozone concentration flowing through this reactor. And we can use that information along with some other information about the system to calculate our deposition velocity at a given time. Okay, so now we're going to have Melissa, one of my students, come over and actually walk through how an experiment is set up.
Okay, so the first thing I do is I would load a sample. And so to do that, I unhook the clamps that hold the lid to the chamber pot. I take out this little stand. Right now the chamber pot's empty. That's just the Teflon lining to help make it less reactive. And we'll run a carpet sample that has been tested and run. But before I would actually take it out, I'd put gloves on. And it's important not to touch the majority of the glove with your hands because the skin oil is highly reactive with the ozone. The carpet sample has been stored in the chamber, which is at roughly 25 degrees Celsius. It's been wrapped in a Teflon film so that way it doesn't get reacted by dust and stuff. And when I load it face up so that the carpet is what's reacting. And then I put the rubber seal around to double check that it's going to seal properly when I close the system back up. And I put the clamps back on. And put the block back under. And then I would come over to the program to load the proper sequence so that I know when it's bypassing and when it's going through the reactor. And today we'll run a 24-hour sequence to make sure that it's all working right. So I would pull it up and then I would reset the date so that it's the proper timestamp. Then the next thing I would do is check the flow rates to make sure that everything's working well with the system. So the first place I check is if it's completely bypassing the system and it's all just going through to the activated carbon.
And you didn't need to show the actual flow check. Just mention the flow. Yeah.
Okay.
And just where you check the flows.
And then I would check the bypass, so I would hook the flow meter up here and put a cap down here so that none of the flow's passing. And then I would set the flow so that's just bypassing the reactor, and then I would check through the reactor. And the flow rate should be about 2.15 liters per minute. And so that's how the system works.
Okay, great. Thanks, Melissa. All right, now that we've seen the experimental chamber and seen how samples are placed into the chamber, we'll take a look at what we do with that, the data that comes from that chamber. Before we do that, let's review the chamber. Here's what it looks like. Just a moment. This is the chamber, this big guy here. And all of this back here is really for controlling the flow into and out of the system. This is a diagram of the system. It's very close to, this is an older diagram, so it's not exactly the way it was set up right now. But I'll run through it just to give you a sense for how this works. It's very, very close to how it's operating right now. And so we have this air feed. We have the air delivered to the chamber through a series of valves, and it can be humidified, as you can see here, and we can add ozone. We have the ability, using these valves and an automated system, to control the flow direction. If we bypass it, as shown by this arrow, then we can check what's going on at the inlet, essentially. What's the inlet concentration? And if we shut that valve off, shut this valve off, and then have it going in this direction through these valves, then we're measuring the concentration at the outlet. This is a sample down here, and all of this over here on this side, let me grab that, is not associated with the chamber as set up right now, but this is for measuring byproducts that might be generated in the reaction of ozone with surfaces. This is what the raw data from the chamber looks like. We run an experiment in which we set it so that we're measuring the inlet concentration first for about 20 minutes, and then we run for several hours, and with it going through the chamber and essentially being in contact with the sample, and then we run for about 20 minutes with it running bypass again, so we're looking at the inlet, and so we keep doing this back and forth, and from this we can get information about the difference between what's going in and what's going out and how much ozone is being consumed by the sample. We'll come back to this in a minute. The way you measure a deposition velocity, or the way you calculate it, is at any given point is by taking the inlet concentration and outlet concentration and putting them into an equation like this. I won't go into the derivation here, but just to note that the volumetric flow rate and the area of the sample come into it. And this is like a quick diagram of the chamber over here on the right. And you can see that the inlet concentration would be higher than the outlet concentration because it's being, essentially, the ozone in this case is being consumed by the sample. Now, this equation is a simple version. It doesn't account for the fact that you can have removal, essentially, of ozone on the chamber itself. And so we do have to take that into account here. And so there's a modified equation that looks like this, in which we can account for the ozone that is removed by the chamber itself. It's relatively low reactivity, but it's enough that we need to keep an eye on it. If it gets too high, because it gets contaminated or somebody touches it with their fingers, that sort of thing, then we have to clean it and make sure it's very clean. But we do take into account the concentration ratio that we would get in the absence of a sample. And the sample, we actually replace the sample with a Teflon sample, and that gives us a essentially a completely unreactive background. And that's our, there we go. So that's how we calculate the deposition velocity. Now let's take a look back at our data. So this is what our data looks like. It's a continuous process. We run for about 24 hours, and we have these initial and periodic switches to the inlet, but most of the time we're looking at the outlet so that the sample is mostly exposed to ozone during that 24 hours at some level. Oh, I needed just to note here that, well, we can go here. If you recall, that equation required the inlet and outlet concentration. So what we do is we take an average of points from this region, that is our inlet, and the ratio of the inlet to the outlet is actually the ratio of those 2 points. We take a 5-minute period right before it switches to the inlet and a 5-minute period right before it switches back to the outlet. And those two give you your inlet and outlet and so what you get is a deposition velocity at a particular time. In this case, it's 6 hours. We also get one at 12 and 24 hours by looking at these regions and this region. Okay? This is how it's reported. You see a sample, it's a carpet sample, this is the sample dimensions, a sample name. We check things like the flow rate and temperature. This happened to be a lower temperature than we normally operate. And, oops. And this is what the data looks like, and down here are the calculated values of the deposition velocity and information about the ozone. And in this case, the average deposition velocity for this sample was 7.4 meters per hour. Putting that in context, carpets tend to be higher reactivity. That's typical. And this is a bunch of data from specific materials that were tested for a project a few years ago. And the codes are wall, ceiling, and floor materials. We do it that way because there's it just made sense at the time. That doesn't tell you whether it's paint or whether it's a vinyl flooring or what it is, but I can explain a few of these. For example, this is a wall material, and this actually happens to be a type of paint that has a collagen in it, and it was actually quite reactive. This one here is a ceiling material that was fairly reactive, the kind of thing that you would have in a drop-down ceiling. And so those are high reactivity things, and you can see that this floor, this is carpet, right there, so it's fairly high reactivity. And some of these other ones, like for example, this is probably, I believe, this other direction, is I believe a clay paint of some sort. Actually, this one down here is a clay paint. But anyway, there's different kinds of materials. On the low end side, flooring actually tends to be low reactivity because the floors tend to be made to be, you're walking on them, you don't want them to absorb things, they tend to have low surface area other than carpet. And so things like vinyl flooring, coated wood materials for flooring, those tend to be very low reactivity and wouldn't contribute much to removal of ozone in an indoor environment. Wall paints, typical latex wall paints, are slightly higher reactivity, but not a much higher. Here and here. This one at the top is an exception, but that was not a latex wall paint. So that gives you a little bit of context for the values of the deposition velocity. And then putting it in context of what these materials would do to ozone indoors. This is just an example for a typical home that has very simple setup. You have some walls that are coated with something, wall and ceiling coated with something, and then floor coated with something, or covered with something. The first one is a carpeted floor with latex painted walls. And what these values are, show you, is exposure to ozone, assuming some information that the outdoor concentration is about 40 parts per billion, air exchange rate is typical, that sort of thing. This red indicates products that are reaction products of ozone with surfaces. You can kind of ignore that for now. The main thing to look at are the white and black bars. They represent your overall exposure. The black bar is the outdoor exposure that you get from being outside. So that never changes. You have a certain amount of time that you spend outdoors, maybe 5 to 10% of your time, and you get a certain amount of ozone exposure from that. The white is actually your indoor exposure, and that depends on the reactivity of surfaces. You can see that it's modest here, and a lot of that has to do with the carpet removing ozone. But the fact that there's carpet there means that you end up with a lot of these reaction products here. If you said, oh, let's get rid of the carpet, put in vinyl flooring, it's not very reactive, we're not going to have those byproducts that we worry about, you end up with a lot more ozone exposure because the surfaces are not removing the ozone. If instead you stick with the vinyl flooring, but you coat the walls with something that's very reactive, in this case I note clay-coated walls, which we know do remove ozone, you can have a substantial removal of ozone, a substantial reduction in ozone exposure if you have a highly reactive wall and ceiling. So that's it for this set of videos, or this video for the deposition velocity. And that should give you some context for how the experiments were done and what they mean, what the values mean in the real world. Thanks.